I'm Daniel Levine, and this is the Bio Report. Cytokines control nearly all aspects of the immune system. As drugs, though, they've had limited potential because of inherent drawbacks. Simcha Therapeutics is engineering versions of cytokines to activate various immune cells in the fight against cancer. Simcha's most advanced program, ST067, is the first in a new class of interleukin-18-based immunotherapies designed to overcome a key defense mechanism that the tumor microenvironment produces to prevent interleukin-18 from doing its work. We spoke to Sanuj Ravindran, CEO of Simcha Therapeutics, about the company's engineered cytokines, how they work, and why they may represent a powerful addition to the immunotherapy arsenal. Sanuj, thanks for joining us.
Thanks, Danny. Pleasure to be here.
We're going to talk about Simcha Therapeutics, its work on interleukin-18, and why this can offer a potential strategy to enlist the immune system in fighting cancer. Before we do that, though, I'd like to start with your decision to join the company in August. What attracted you to Simcha? What did you see as the opportunity?
Yeah, well, actually, I joined the company in May. We put out the press release in August, but I've been with the company since May, and it's been going really well. I first got attracted to the company really based on the science. We're in an area of novel biology in an industry where a lot of work, is incremental. It's really great to be in an environment where we're working on a really novel area of biology that has really transformative potential. And what I mean by that is the biology around IL-18 is something that is really elegant and intuitive in terms of the sense that it makes. And the work that our founder has done to better understand that pathway and therefore now knowing how to drug it effectively is a recent revelation. And we're excited to be really the first in class, first in the clinic with a product that can deliver an potentially effective IL-18 therapeutic. So it's novel biology that makes intuitive sense, that has really, the potential to make a meaningful change in the lives of cancer patients, and for us to be first in class and well ahead of others is a good spot to be in. So those are the first things that attracted to me. And then, of course, as I got to know Aaron and the team and the investor base that we have, all that really came together very, very nicely.
IL-18 is a cytokine. Remind listeners what the role of cytokines are within the immune system.
Yeah. So you can sort of think of cytokines as, cytokines are to the immune system as hormones are to the endocrine system. So really cytokines are proteins that allow immune cells, such as T cells and natural killer cells, to communicate with one another. So they're messenger cells. And that aspect is really, really critical when it comes to orchestrating the body's defenses to fighting diseases such as cancer. So if you think of cells like T cells and NK cells as doing the killing, cytokines are the proteins that direct the message and direct the killing.
In the case of certain cancers, what happens to interleukin-18?
Well, interleukin-18 is a very interesting cytokine because it acts both on the innate and adaptive lines of defense from an immune standpoint. And when interleukin 18 is given as a therapeutic, there are a number of reasons why it is an optimal cytokine. But what happens in the face of cancer, particularly within the tumor microenvironment, is that cancer cells induce the expression of a protein called IL-18 binding protein. And IL-18 binding protein has on it a decoy receptor that effectively binds all of the IL-18 that prevents it from reaching its intended targets on cells such as T cells and NK cells. So that is one of the, so you can think of it as sort of an immune checkpoint in that sense, preventing IL-18 from effectively carrying out the messages to its intended targets.
It had been theorized for some time that you could potentially take recombinant IL-18 and use it to treat cancer. What was the case for doing that?
Well, the case for doing that is the biology around how IL-18 works. So IL-18 acts on tumor infiltrating lymphocytes, basically antigen experienced T cells, and not all T cells, really just those that are antigen experienced. So they're not going to indiscriminately, hit T cells more broadly than you need them to. They act on natural killer cells, so that's the innate immune system, and it also acts on regulatory T cells as well. It downregulates regulatory T cells, so it has sort of a trifecta of effect in terms of how it works biologically. So theoretically, it's a very optimal cytokine in that sense. And also, unlike many other cytokines, IL-18 acts as more of an amplifier than an on switch. So concerns around tox, for instance, are minimized somewhat because of that aspect. And because it also acts as an amplifier, IL-18 can be paired with other modalities. A, because it acts as an amplifier. B, because it can act to drive T cells, to drive NK cells. You can compare this potentially with other checkpoints, other cytokines, certain cell therapies, certain bispecifics. And so it has great potential as both single agent therapy as well as in combination. So those are the reasons, one should theoretically be excited about IL-18. And in fact, IL-18 had been taken, recombinant IL-18 had been taken into the clinic by another company in about 12 years ago. And it was studied in a phase 2 study. And unfortunately, in that study, while the drug was well tolerated and shown to be safe, it wasn't very effective. The sponsor of that study only saw one partial response after treating 63 patients. And so the decision was taken shortly after that. The results of that study came out to pause the program. So this particular sponsor paused the program, and I think it put a damper on the program in general in terms of others looking into it. So it has been taken to the clinic already. What was later then discovered, and this is where the scientific work of Aaron Ring and others has really been effective at elucidating the biology around IL-18. What was later discovered is that as IL-18 is administered, and as I described a few minutes ago, this IL-18 binding protein is overexpressed, and effectively the recombinant IL-18 that's administered isn't getting to where it needs to get to because it's being taken up by the binding protein. And so what Simcha Therapeutics has done, or what Aaron Ring had done out of his lab at Yale, is develop a variant or an engineered variant of IL-18 that we call decoy-resistant IL-18 that is engineered to avoid IL-18 binding protein so that it can get to its intended targets.
So how does it actually get to its intended target?
It's using the natural receptor on T cells and NK cells, and so it's not doing anything different in terms of how it interacts with its receptor partners. The one thing that it's doing different is avoiding the binding protein. So it's the binding protein that was basically intercepting the wild type 18 and preventing it from getting it to its intended receptors. We avoid that interception and get to the intended target, interact it with it the way it otherwise naturally would.
Your lead experimental therapy, ST-067, activates both the innate and adaptive immune system. How exactly does ST-067 work?
Well, we're in the clinic right now, so we're in the process of finding all that out, right? In terms of how it works biologically, it like you said, it acts on both the innate immune system, which means that it interacts with cells such as natural killer cells, and it acts on the adaptive immune system, which means it interacts with cells such as T cells. So the way IL-18 works is that it's administered. In our case, we're administering it subcutaneously, and it interacts with those intended cells, and then it programs those cells to then engage in the killing of cancer cells. In terms of how it's working in the clinic, we started a phase 1 study in late last year. We're in the midst of that study right now, and we're continuing to advance the drug in that study. And once we complete that study, we'll be able to say more about how the drug is doing in terms of biological activity, safety, efficacy, as well as overall tolerability as well.
What indications are you pursuing?
Well, at this stage, we're looking at a variety of indications. So the first step for us is to have taken this into a single agent dose escalation study in a variety of checkpoint refractory solid tumors. So these are all patients that have a variety of solid tumors and these are patients who have received other checkpoint therapies and have not responded to those therapies and are now entering our study as the next stage of their treatment course. And in that, we are looking at a variety of tumor types. And at the conclusion of the study, we'll have a better sense of which tumor types are likely to be better responders versus others. And then that will guide the development program going forward. And at that point, we'll hone in on more specific indications.
Is there any sense how broadly applicable this might be?
Yeah, I mean, we think this could be very broadly applicable. We're starting with tumors that are known to be more immune sensitive. And those are tumors like your melanomas and your renal cells. But we're also including tumor types that are less historically immune sensitive, because we think that because we act on both the adaptive and immune pathways, that we think that we have the potential to address both hot as well as what they call cold tumors. So we don't know for sure yet whether that is indeed the case, but theoretically it could be. And that's what we're interrogating right now. So in that sense, it could be broadly applicable across many tumor types. We are looking at this as a single agent therapeutic. We are also planning to look at this in combination with other modalities. The first of those would be in combination with the anti-PD-1s. And then beyond that, we are evaluating and prioritizing other modalities with which IL-18 could be a good therapeutic partner.
When you say other modalities, you're thinking of things like TKIs or other immunotherapies?
Um, a variety of modalities, including other immunotherapies. So the first one is another immunotherapy, right? So the PD-1s are immunotherapies. That's naturally where one first looks to look at combinatorial effect. Beyond that, we are looking at other novel combinations, and that could be in a variety of areas that we haven't fully disclosed yet.
What's known about the safety and efficacy of the drug to date, and how specific is it when you elevate levels of IL-18?
Yeah, unlike other cytokines that have a lot of what they call pleiotropy, IL-18 should be relatively specific because we act only on antigen experienced T cells, not more broadly than that. And we act, as I said, on NK cells as well. And so we think that we're, from that standpoint, have a better safety profile potential than other cytokines. In terms of the experience with the drug, as I mentioned, wild type IL-18 has already been in the clinic, including a 63 patient phase 2 study in which the safety profile was relatively favorable, especially when you compare it to other cytokines that have been in the clinic. So from that standpoint, we know that we're looking at a relatively favorable safety profile. From the specificity that I mentioned earlier, we feel good about the safety profile from that standpoint as well. And to date, we're pleased with what we are seeing in the clinic. We started the study a year ago. We're marching through multiple dose escalations that we've been able to move forward with. We haven't said much specifically about where we are in the study, but it is progressing nicely. And from a safety and tolerability standpoint, looking favorable thus far.
You're focusing at first on non-responders to immunotherapy. Is there any way to predict which patients would likely be non-responders and be better off using an IL-18?
Yeah, that's a part of the phase 1 program. We are collecting the translational data, if you will, that will allow us to potentially identify those markers that will help us potentially identify those patients who might be better responders or those that are less likely to respond and why. So that's all part of the current clinical study.
Simcha raised $40 million in a Series B financing in January. How far will existing funding take you, and what's the plan for funding the company from there?
Yeah, we've raised $65 million in total, including the most recent Series B that was led by SR1. We also have other very notable investors that are with the company. Those include investors such as BVF, Samsara, Logos, Aramark, Rock Springs, in addition to our early investors, Wuxi and Sequoia. So we're very blessed and privileged to have a great group of supportive investors. And fortunate, to have raised that round, very recently, because in today's current environment, I certainly wouldn't want to be out raising capital. We have we're in a good position. The expectation is that financing will allow us to complete this phase 1 study, start the next combination study, and take on a few other activities that push our runway well into 2024. And so we feel like we're in a good position to really put our heads down and execute on the current clinical plan. And in terms of next financing, we still have a fair bit of time before we have to embark on a formal financing effort.
Sanuj Ravindran, CEO of Simcha Therapeutics. Sanuj, thanks so much for your time today.
Thanks, Danny. I really appreciate it.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.